Good morning, everyone. The topic for today is the dihybrid cross. But what is a cross? And how do we make predictions? Let's move on to, before moving on to the topic, we should know that some of the traits come from the mom's side and some of the traits come to us from our father's end. As we know, there are 23 pairs of chromosomes in human beings. Similarly, in all the organisms, these chromosomal pairs differ, but the chromosomes exist in pair only. So, during the process of cell division, in the reproductive cells, these chromosomal pairs get separated and the gametes which are formed are consisting only one allele of the gene pair. So like we can see, both mom and dad have 23 pairs of chromosomes. They inherited one from each of their parent. To make up the next generation, the DNA is mixed up for the genetic diversity. So each mixed up chromosome goes up to its own sex cell. The next generation gets one chromosome from each parent. The baby has its own combination of parents' DNA. So our problem for today, the task for today is this question, that a pea plant that is heterozygous for round yellow seeds is self-pollinated, self-fertilized. So what are the phenotypic ratios of the resulting offspring? So the first step is to determine the parental genotypes. So first of all, we should understand what does the word heterozygous means. It means that it contains two different gene alleles. And another clue given in this question is that it is self-fertilized. So it will be crossed with itself. So both the parent will be having the same genotype. Next, we need to determine the gametes. The gametes are determined by keeping one type of each allele in combination with each other trait that we are keeping. So we can make as many combinations as possible. So we combine all the R's and Y's to represent the sperm and egg. So for both the parent, it will be capital R, capital Y, capital R, small r, capital Y, and small r, small y. In the third step, we need to make a table with as many rows and columns as the genotype of each gamete that we had found out in the previous step. So out of all the combinations, we could make 4 possible combinations of the gametes. So we will be needing 4. 4s are 16. So we will be needing 4 columns and 4 rows for this particular question. All of you are required to make this table, and we will be writing down the male gametes on the top of the box, outside the box, over the columns, and the female gametes on the left side of the box over the rows. Then we will be making the combinations like this. we will see which row and which column are clashing, intersecting, and that particular combination is to be written. That is very easy to write. Like here, if this is a different question, different combination, I have not put up the correct answer for you because that you will be doing for yourself. So this is just an example that I have posted. If we see the intersecting block of the first column and the first row, we see that it is capital R, capital Y, and small r, small y that are meeting in this block. So we will write capital R, capital Y, small y, but the gene pair, the gene alleles are to be kept together. So we will write capital R, capital Y, small y. Similarly, for the next column, we see that the intersection brings in the capital R, small y, and small r, small y, and so we make the combination. Making a dihybrid cross is a more complex step, so let's understand the simple ones first, then you will be able to do the complex ones also, and you will enjoy it also. So the table or the blocks that we are making, this particular square is called Punnett square. So it is a tool to predict the percentage of the genotypes for the offsprings of two known parents. The genotype of the parents is known, then only we can find out the possibility of the genotype of the further generation. So each square represents a fraction or a percentage of the offsprings with a particular genotype. Like here in the square we have 4 sections, so each section depicts the 25% or 1/4 of the offspring's nature, genotypic nature. So we will make a box like this and divide it into 4 small boxes. We are dividing into 4 boxes because we are considering only one trait as of now, because I feel that you are still not clear with the single trait also. So when we are considering single trait, it is a monohybrid cross, and we are considering the 2 traits together, it will be a dihybrid cross. Then we write the male parent's allele along the top. Like here we see capital R, small r, and the female parent alleles along the left side of the box. Right, it is written in blue here, capital R and small r. This was the case of the self-pollination. If the self-pollination is not occurring, or we are considering the parents, homozygous parents, so we consider that the one parent is completely dominant and having both the gene allele dominant, and the other parent is having both the recessive gene allele. So then also we write it the same way. The male parents alleles are written over on the top of the box, and the female parents alleles are written on the left side of the box. So if we consider a simple example of the pod color, green pod is dominant over the yellow pea pods. So here we can see capital G, small g. Because it is colorful, you will be able to get it more clearly. Small g and small g. We will see where the these letters are intersecting. Like in the first block, the small g and the capital green G are intersecting. So we will write small g and the capital G. only the capital letter will come first. We will never write small g and capital G like this. Okay, so in the next block also, right one, it is capital G and small g. Down we have small g, and down right we have again small g, small g. Now to write their genotypic ratio, we will see how many type of gene combinations we have got. Like here we have 2 capital G small g, so it will be 2:2, that is 1:1. So there are 50% chances of the pod being green and the 50% chances of the offspring with the yellow pod with this combination of genes. So why Punnett square? Why can't we do it like this? A Punnett square actually allows us to do our combinations, make the combinations very systematically without any mistakes. If we write random combinations, we will somehow miss out something or make some wrong combinations. So this will be the answer of your question that we had started off initially. The self-pollination of dihybrid traits of round, yellow or wrinkled green pea seeds. So you can check your answers. I'll send this image also, and it is also giving the ratio of the offsprings with the possible particular genotype. The color of the picture shows the phenotype of the offspring. So you must have understood the difference between the genotype and the phenotype, and also which ratio of the offspring will be showing the particular phenotypic character. Like out of 16, 9 seeds will be yellow and round, 3 will be green and round, 3 will be yellow and wrinkled, and 1 will be wrinkled green. And lastly, we need to write down the genotypes of the offsprings and summarize the genotypes and phenotypes of the offsprings, so that will make our answer final. Without the summary, there won't be any use of your Punnett square. So now give yourselves a good practice, and you can ask your doubts if any.